Good afternoon, everybody. Good morning, good night, good evening, wherever you are. I welcome you on behalf of ISTA and ISSS to our webinar on seed dormancy. We have two speakers today, and the first one will be Ganesh Jaganathan, with the title, The Ecological Significance of Physical Dormancy from Conundrum to Clarity, giving a little bit background on this topic. And the second presentation will be done by David Johnston on dormancy breakage for germination testing. I welcome both speakers here. Before we start, just an information for everybody. This session is recorded and we will have it posted on the ISTA YouTube channel a little later. You can look at it there. In between the two presentations, we have the first Q&A session. To post questions, please use the question box to pass your questions on, and I will then ask these questions to the presenters. If there are questions we cannot answer, we will pass them also to the presenters, and you might get an answer privately. Thank you for that. And with that, I would like to start with our first presenter, Ganesh Jaganathan. He is an associate professor in Germplasm Conservation Unit, Institute of Biothermal Technology, University of Shanghai for Science and Technology in Shanghai, China. Very nice place. And his research interests include germination, ecology of recalcitrant seeds, cryopreservation of difficult to store seeds, dormancy, seed germination in changing climate, and soil seed banks. Ganesh, I pass the screen to you to start your presentation, and each presentation will be about 30 minutes.
Wonderful. Thank you so much for that nice introduction, and thank you for inviting me to deliver this topic, which I've been trying to get it out for quite a while now. So the topic, as you might have known now, is moving from conundrum to clarity. Before I get into the real topic, I'm gonna sort of like introduce into the brief background of why this is kind of like a confusing area and how we can move towards clarity. All right, so seeds have been studied for a long time, and dormancy is one of the key aspects that we picked up at least 200 years ago. And there are some other languages that showed dormancy is an occurrence even 500, 600 years ago. But the first solid paper that showed dormancy is a mechanism to sort of like blend in with the environment is the one by Crocker in 1915 or 1916. And after that, there are so many attempts to make some sense to what this is. In particular, the work by Baskin and Baskin is incredible, because they try to classify the dormancy into 5 broad classes, and in some other languages they call this as a sleep. And I like this word for 2 reasons. Number one, as humans we sleep, and sleep is the time where our brain is more active. So dormancy is the time where the seeds are more active. So the 5 classes I talked about, they are classified into morphological, physiological, physical, and combinational. And in the past 50 years or so, we have come a long way to understand each dormancy type, and now we have a solid understanding that morphological and physiological dormancy, which by the way is having a underdeveloped or undifferentiated embryo is a morphological dormancy, and physiological has something to do with the embryo or the seed coat, and in physical, the seed coat is impermeable, which I will talk about. Most of my talk today will concern physical dormancy, and combinational dormancy is a combination of physical and physiological. So we now know morphological dormancy and physiological dormancy can coexist with other traits. For instance, desiccation sensitivity or recalcitrance is a very common, and we thought recalcitrance and dormancy can never co-occur, but recent evidences show the opposite. So even epicotyl dormancy is more in morphological or physiological dormant species. But when it comes to physical dormancy, I'm going to just restrict my topic to physical dormancy, my bad, they don't co-occur or coexist with any other traits. So as I just mentioned, physical dormancy occurs due to impermeable nature of the seed or fruit coat. So I have had many occasions where many people would argue physical dormancy could also occur due to endosperm, cotyledon, and embryo. In fact, they would have little to no role in physical dormancy. But these structures, they do influence physiological dormancy when you have a combinational dormancy. All right, so one thing we have to bear in mind is physical dormancy would never occur in any recalcitrant seeds. I know this seems counterintuitive, where some papers claimed to have, or at least inferred to have, physical dormancy in recalcitrant seeds. And I have shown you a nice example. This is from Fagaceae. This is one of the largest family where dormancy has not or had not been very widely studied, but now people are showing a tremendous interest into this, and some of the people are even trying to conclude the recalcitrant seeds of Cucus or all the Fagaceae family itself, they have physical dormancy. So in a paper, I tried to clarify this. So that was in 2021, where I tried to say, recalcitrance and dormancy are not necessarily mutually exclusive traits, but physical dormancy and recalcitrance are definitely mutually exclusive. So one more thing before I get into the nitty-gritty of my talk: physical barrier is not equal to physical dormancy, and many people, they try to associate these two words. It could be because of the physical, I don't know what is going on. So for instance, in Lauraceae, as we showed, Lauraceae is an interesting family where some species have physical dormancy, others have recalcitrant, and others have orthodox, and they are water permeable, meaning the water could go in through the seed coat, but in others, the water would not. So the only way to confirm physical dormancy in a species is by conducting an imbibition test. A subsequent question would be, how long do I have to carry an imbibition test to confirm physical dormancy? A reasonable amount of time, I would say, is 30 days, because that is the sort of like a threshold period. If the seeds had no physical dormancy, they would have observed water. So water absorption is not so obvious when you visually inspect the seeds. So the best way is to sort of like weigh them at a regular interval and then compare the weight with the control seeds. So I think I kind of like finished talking about the basics of physical dormancy, and now let's have a look at what we have done in the past 5 decades. So in a nutshell, we have studied 3 areas intensively. So the first one is how the environmental conditions would onset physical dormancy, or how the dormancy would break. So some of the factors people have looked over the years include rainfall, temperature, humidity, and even the temperature fluctuation. The second important area, I believe, that received nice attention was what structures are involved in opening the water gap, or what structures are during a dormancy break, and we call that as a water gap. So we understood lens in Fabaceae. So that is a common sort of like ground, and we have moved now our understanding to understand almost all the families. We now know a little bit about what the water structure or water gap would be. The third one, which is very interesting, as I just mentioned, is how physical dormancy associate with other traits such as desiccation, sensitivity, and predation and defense. So physical dormant species would invest little towards their defense. So that is one nice advancement we made. Okay, so what I'm going to talk from now on is gonna be a little bit controversial for others, but I hope I'm gonna justify my thoughts on this, and I welcome any questions you may have, towards this talk, towards the end of this talk. Okay, so during development, what would happen? Physical dormant species, any species with a history of physical dormancy, they somehow the seed coat would become impermeable first, and then the micropyle and hilum would remain open. So this is a slow process, and finally at one stage all these structures would close, and that would onset impermeability. The opposite would happen during the dormancy break, wherein the water gap, which would be adjacent to these two structures, micropyle and hilum, this is for Fabaceae, but the process, I believe, is similar for all the families. So the water gap would open, and then the water would enter through this water gap, and then it would hydrate the seeds. The seeds would germinate immediately, most likely within 5 days. So there are 4 possible conundrums I'm going to talk about today. And the first one lingers around evolutionary origin, and the second one is primary and secondary dormancy definition. I hope some of the ISSS members take note of this and try to solve this. The third one is global distribution, how widespread and where physical dormant species are distributed. And the fourth one is dormancy cycling, or subdividing physical dormancy. So most of my talk is going to highlight the fourth point, which is to subdivide physical dormancy. Okay, so moving on, conundrum number 1, evolutionary origin. So in my literature review that I tried to work out during my PhD, I came up with these 4 factors, where there are some people claiming fire acted as an evolutionary driver for the evolution of physical dormancy or impermeable seed coat, and endozoochory, which is when the seeds go into the intestine and the seeds have to survive that acid environment, and that was one of the drivers for the evolution of impermeable seed coat. And recently we have crypsis, which essentially says the impermeable seed coat had evolved to somehow hide from the rodents. And the fourth one, which is an interesting one, is to protect during persistence. So after the seeds disperse, they would sort of like stay in the soil for a long time, establish soil seed banks, and physical dormancy is a mechanism they use to protect during that persistence time. But there are these supporting studies are also criticized by some of the studies, and I would leave it at this point and then move on to why. If you can see, most of them I have contributed significantly to say these are not, or these might not be, the evolutionary factors. And the argument is very simple. If you take any physical dormant species, a tree producing physical dormancy, the number of permeable and impermeable seeds would vary year to year. So meaning 1 year, all the seeds could be permeable. Year number 2, all the seeds could be impermeable. Year number 3, permeable. Year number 4, impermeable. So it can take that cycle. And it did not actually explain, and in particular, crypsis, failed to explain why pine seeds, they have no impermeable seed coat. And if they have evolved somehow against all those factors, what is the need for long-term persistence in physical dormancy, physical dormant species? So this kind of like runs into the thought that these factors, which I just explained, they could be a chance event. And the same applies to all the factors. This is not just for crypsis, but this is a nice example. So going with fire, I can clearly see the interest in fire because of many scientists working on, proving that fire is an evolutionary driver for everything. Even for the plant evolution, they are all over the world, and I can clearly see why they want to put physical dormancy into this basket. And what they sometimes underestimate is sometimes they even include the aerial seed banks as physical dormancy. But as I just mentioned, physical dormancy, you have to have the seed coat impermeable, which is not the case with many of the aerial seed banks. And fire, is not necessarily a dormancy breaking factor in all the parts of the world. So if one would accept, for the aerial seed banks to, the serotiny to have a long fire interval, and they can, after 100 years, they can fall down, and the same would happen for physical dormant species. So the fire interval would be much longer. So that sort of like argues against fire as an evolutionary factor. Okay, so moving on, what if those four were not the evolutionary factors? What could be? So the most logical explanation for the evolution of physical dormancy, impermeable seed coat is the climate change. Oh, sorry, climate drying, climate change, and then the climate becomes drier. So I think Baskins, they did a nice sort of like study where the non-dormant seed would become a physical dormant seed, and then that was about 50 million years ago, 43 million years ago to be precise. And so this is a climate. And then if the climate changes, you would have a new trait, and that could be impermeable seeds. But what about protection after dispersal? This also falls into the same problems with other factors. So I'm going to leave with this quote. So what essentially is happening, I'm going to show a picture of what this one says in a minute. So imagine if you have a wet environment, the seeds, although they can produce physical dormancy, they would never disperse the seeds in an impermeable state. Whereas plants growing in a semi-dry, they are partly dry, and sometimes they are wet, having some rainfall. They have both non-dormant seeds and physically dormant seeds. Plants in a dry environment would always have physically dormant species if they have a known history. So most of the fire occurs in the drier areas of the world. I'm saying most, not all. So that could be the reason why people tend to associate fire and physical dormancy. They go hand in hand. So this is a complicated issue here. If you move a plant from one environment to another, they would lose its ability to produce physically dormant, and for the reasons I'm going to talk about in a minute. But before that, this is the most concrete evidence of how physical dormancy could have evolved. So this clearly says, regardless of the ancestral state, whatever the ancestral state would be, physical dormancy is definitely a derived trait, most likely to have evolved from physiological dormancy. All right, so to resolve the conundrum one, I am proposing climate change or climate drying as the evolutionary factor. And the reason is fire would have acted only on a smaller area. The same would go for endozoochory or crypsis. They won't act on all the seeds. All right, so I'm going to quickly move on to conundrum number two. So now it's time to talk about how do we define dormancy. So most of the people are, in fact, most of the seed literature, we classify dormancy into primary and secondary, meaning primary dormancy, they have the seeds that are dormant at the time of dispersal, and secondary dormancy is kind of like they go into the cycle of becoming non-dormant and then dormant. There was a study published in 2019. They mentioned there are no cases or no known cases of non-dormant seeds becoming dormant after dispersal. So in other words, if you have to have primary dormancy for the seeds to enter secondary dormancy. And the paper was very clever. They never mentioned anything about physical dormancy. I at least get the assumption that they restricted their definition to physiological dormancy. So then I went on to understand this in physical dormancy. So what happened here, typically this is for almost all the seeds, but in physical dormant seeds, you have the seed formation stage and seed maturation stage. And then during maturation drying is when the seeds would become impermeable. So if you take the seeds here during the middle arrow or the left arrow, the seeds would still be permeable. So this created a lot of confusion whether some species even have physical dormancy or not. But to understand that, you have to let the seeds to dry and achieve a reasonable moisture content. So we did some experiment in 2021, and what we did was we collected the seeds early and buried some of the seeds under the plant or under the tree, and some of them, exposing directly to the sunlight. So what this did, the sunlight would evaporate the water, resulting in drying. So these seeds were permeable. These seeds were also permeable because we collected early, around this time, middle stage. And these seeds remained as permeable, and they germinated, in fact, pretty quickly. But these seeds, they got drier, and then they became impermeable, meaning the non-dormant seeds, at the time of dispersal, they were non-dormant, and then they became dormant. So this is another experiment, an ongoing experiment, where we wanted to now understand if there could be any variation between the location of the seeds maturing. Sometimes the seeds could be exposed to the sunlight, where they get the chance to dry more, versus the seeds that are staying inside, where they are, the canopy kind of like gives the shade. So when in our preliminary results, we could clearly see the seeds that are exposed to sunlight is physically dormant, but the seeds that are inside or under the canopy, they did not develop physical dormancy. So that explains why sometimes when we collect the seeds from a tree or a group of trees, you would have some seeds that are impermeable and some seeds that are permeable. So to understand this, I already have some evidence from lotus, and lotus is an interesting species because for those who are arguing fire could be an evolutionary factor in the evolution of physical dormancy, lotus is an aquatic species and they have physical dormancy. So what we found was, if you dry the seeds, you take the permeable seeds, you dry them, when they have a moisture content of about 18, they are permeable. 14, they are permeable. 12, they are kind of like permeable, but you can still see a few seeds becoming impermeable. But right about 10% moisture content, a significant amount of the seeds, or the significant proportion of the seeds, became impermeable. And by the time when we dried the seeds to 8% or so, all of them became impermeable. So what I hypothesized was, at this stage, there could be some seeds that are high in moisture content, and there could be some seeds that are low in moisture content, explaining a part of the seeds being impermeable and the other permeable. So we did the same in some of the species from tropics, and we were able to get the same results. All right, so I'm going to quickly go to resolving this conundrum too. So I'm not sure if physical dormancy is a requirement for the development of secondary dormancy, but if a term enforced dormancy has to be used, use it. Why not? If not, we have some refinement in sort of like defining this. All right, global distribution. I'm going to be quickly going over this one. So these two papers, I don't want to read this completely, but what they proposed was using Fabaceae as their study species. They collected as many information as possible, and they said physical dormant species or physical dormancy occur predominantly outside of the tropics, and they went further and said physical dormancy may be disadvantageous in tropical habitats. But I don't have to take anything with this one. There are so many studies. If you look at the paper by Jayasuriya, out of 100 species, Fabaceae, 86 had physical dormancy. I don't know how these two studies missed that, but whatever that is, I think they work with the global GBIF database, which are poorly represented in the tropics, especially from Africa, South America, so that could have contributed to this misleading information. So I did a reanalysis of the data of the information presented by Baskin's book 2014, and this is what we see. We can clearly see tropical ecosystems have more physical dormant species. Okay, so physical dormancy, I would argue, is more dominant in the tropics. Moving on to conundrum number 4, I'm going to talk about this cycling of physical dormancy. So when you dry the seeds, this is where the seed forms. You have a high moisture content. These are arbitrary. So the moisture content would decline, and then at some stage, the moisture content would tend to do something at about 20. This moisture content may be arbitrary. It could be species specific, but about 10, it would become a shallow dormancy. So let's say from 10 to 4, it's shallow dormancy, and anything below 4 is absolute dormancy. So understanding physical dormancy with this shallow and absolute based on their moisture content is very important, and I'm going to tell you why that is the case in a minute. So this is a sort of like a conceptual model how this one would go. So you have shallow dormancy here, and then this would go in sequence until they open the water gap and germinate in the growing season. And any arrows means we don't know. That is kind of like a hypothesis, which future studies are expected to shed more light on this. Okay, so I maintain dormancy cycling is different from sensitivity cycling, and because of the time, I'm going to sort of like quickly move towards clarity. So one of the fundamental questions that got me hooked up when I did my PhD was, you store the seeds at room temperature. You have all impermeable seeds. You store them at room temperature. After 1 year or 2 years, 10% would become permeable. And the second year, you would have another 10%. So this question kind of like bugged my mind for a long time. And then slowly we came to a realization that this could be due to the moisture content variation between the seeds. If you have 100 seeds, each seed would have a different moisture content. And going back to this model, some of them would be in shallow dormancy, some of them would be in absolute dormancy. So these seeds may come out of dormancy early when you store them at room temperature. And more importantly, the same pattern might continue with summer temperature and up to the fire temperature. However, the temperature above that would kill all the seeds. So there is a nice sort of like connection between the temperature, moisture content, and the duration required for the seeds to come out of dormancy. So I said moisture content would influence the dormancy break, but are there any evidence? So in this paper, 2016, I tried to propose this as an idea, and I hope, at that time I was not able to study this in detail, which we are doing now at least, but the nice evidence was presented by, by some scientists working with fire. And I happen to see this one, and this result, seeds with elevated moisture content have lower lethal temperature thresholds, leading to increased seed mortality during fire events when seeds are moist. So what essentially they are saying is the higher the moisture content, you have a different pattern going on. The lower the moisture content, you have a different pattern going on. So this is one of the area we are working intensively. I am now trying to understand if moisture content plays an important role. The seed coat would have a lot of fat, so the association between fat and water would be the major force for this pattern to occur. So in other words, seeds with shallow dormancy would have more water so that they can interact with the fats, all the saturated and unsaturated fats, and also the drier the seeds, they have limited freezable water to for the interaction. So this would likely affect the way the seeds would form dormancy and also break dormancy. So there are, when viewed in this perspective, there are so many questions still remaining. So the water gap would open only during the seasonal temperature fluctuation and fire. So any other means that we know of would crack the seed coat. So for instance, liquid nitrogen, boiling water is similar to fire, dry heat is similar to fire at that temperature. So any other temperature like liquid nitrogen, acid treatment, hydrochloric acid, sulfuric acid, whatever we do, we are not actually opening the water gap, but we are making some cracks everywhere. So we have to know why this water gap opens, or what is that changes during that dormancy break that leads to this water gap opening. So water gap is a different structure than other parts of the seed coat, but what is that one thing that the seeds have to have the trigger so that the water gap would open, and we have no idea what that is. So another question I am going to study more in the future is sort of like water gap complexes. There would be 2 or more water gaps opening. So why? What is the significance? Does it have any relationship with water content? Does it have any other mechanisms going on? So that is one of the things we are investigating. So not necessarily you can expect every seed to open the water gap. So it depends on the treatment. If you put the seeds in sulfuric acid or other acids, liquid nitrogen, they would more likely have a gap, which is not a water gap, but a crack. So like I said, the seeds would germinate immediately, so there is no reason why they should not. So they have to time their germination to the most optimal time so that they can grow and establish seedlings. So now, because of the moisture content, like I said, because moisture content is an important component in understanding physical dormancy, which somehow people missed before. How can we measure the moisture content in physical dormant species? So this is a question open to ISTA and also ISSS, people who are working on moisture content. I can clearly see there are so many methods. We recommend grinding the seeds, but in my opinion, if you put the seeds at any of these higher temperatures, they would open the water gap, and that would behave like a normal seed. So I don't know, we need some clarity along this methodological problem. With that, I'm going to summarize what I talked, but I would not do that because of the time. So I'm going to thank all these students who worked hard to do some of the brilliant works in my lab. And with that, I wanted to conclude my talk by saying this. So I was reading this paper in particular, which was more similar to the problems we have discussed. So in an informal and unpublished presentation to the British Ecological Society in 2004, Peter Grab expressed some dissatisfaction that progress in ecology was often hampered by our failure to reject wrong ideas and faulty interpretations, which rings bell with physical dormancy. I agree with this conclusion and suggest that this problem is in part a consequence of specialization and fragmentation of ecological research. So essentially what they are saying is, if you have so many, ecology with its numerous subdisciplines can sometime resemble an amorphous post-mortem or, so there are so many theories or so many wrong ideas which would drive the science in probably not the right way we would expect. So I hope I have given some clarity, and thank you so much for this wonderful opportunity. And I'm happy to answer any of the questions that you may have. I'm sorry I'm a little bit late, but yeah. Thanks again.
Yeah, thank you, Ganesh. I don't have any questions yet, but we might get a few. Please use the question box to put your questions in. In the meantime, I didn't introduce myself. My name is Andreas Weiss. I'm Secretary General of ISTA. Today, not talking from the headquarters in Switzerland, but from the World Seed Congress in Cape Town. In between, while we're waiting maybe for some upcoming questions, I can tell you that the ISTA annual meeting was last week in Verona. We had a nice seminar there as well on biodiversity, and you can have all the presentations on the ISTA website, ctest.org. You will find them there. I would also like to invite you on behalf of ISSS to the 14th ISSS Biennial Conference, which will be held at Sorbonne University in a few weeks' time from 3 to 7 July. And if you want to know more about plant dormancy, there's a plant dormancy symposium in September 12 to 15, which will be held at University of Western Australia. Not the closest way, but now we have a few questions coming in. That is good. So we took the time to get some questions. A technical thing, please share the presentation slides. You can find all the presentations in a few days' time on the ISTA YouTube channel. We are not sharing the slides, but you can have a look at all the presentations again. I have here a question from Marie-Helene Wagner. Have you ever met or read about physical dormancy on brassica species?
That's an interesting question. Not really, no. Because yeah, I can see. I don't know who asked this question, but I can see this one is kind of like a tricky area where some people would try to claim a family has physical dormancy, but I have no idea if this family, would have physical dormancy. I don't. I'm sorry.
Good, and that's more a compliment to you, Ganesh, from Sergio Pasquini. Your speech is very clear at the level of Finch Savage.
Okay. So, that's a compliment. Thank you.
That's a compliment, he says. And there's a question for you. I would like to have some more information about ecological dormancy.
Yeah, so so as I showed, I think that is where I introduced the dormancy briefly, so that would help some others to have a look as well. So epicotyle dormancy is an interesting dormancy, which we see it is in the seedlings, not necessarily with the seeds. So that is a mechanism some of the seeds would use. So they germinate quickly, and then they have a pause in their growth. And then, it could be during the winter or it could be during the summer. And after that, they would continue to grow, which is in contrast to seed dormancy, where we have seeds would stay in the soil and they would do. But whoever sent that question can send me an email. I will send a lot of literature about epicotyl dormancy.
Okay. Another question coming in, could you use ERH, which is equilibrium relative humidity, as well as oven drying?
Yeah, so that is for the moisture content estimate, right? But over the time, I figured out, because when I was at MSBP, they did this equilibrating ERH. But the problem is, when you dry the seeds, they would physical dormant species would become impermeable. So after that, I don't think ERH would dry the seeds if you wanted to dry. So to find that moisture content, there would be some hiccup. You can still do it, but I don't think that is the right way. But oven drying is the best way. But I'm not sure if you have to chop the seeds and do it. That is where my doubt is too.
Okay, I go to the next one. We still have some time to do so. Let me go up to the next question. Is damage caused by high temperature, lethal temperature in brackets, related to the moisture content, related to ROS species formed from water molecules?
It would be, definitely, because when the seeds have higher moisture content, they would not have that lethal temperature reaching there. So that is what we are trying to study now. So what would happen if the seeds are drier, they would behave, if you throw them into a fire temperature, they would behave differently. If the seeds have slightly higher moisture content, if you throw them into the fire temperature, they would behave differently. So moisture content would definitely influence that, yes.
I got another question here, first, also a thank you for the nice presentation. We'll contact you through email. That is from Samir Abekar, but there's also a question related to that. It would be interesting to know if you share info about bittergourd dormancy.
Sorry, what dormancy? Epicotyl?
Bittergourd.
Oh, I see. Okay. Yeah, I have done some work in the Southeast Asia, where we do see physical dormancy. If you dry the seeds, you do have physical dormancy, although it is not very clear what moisture content they would have that. But we do have some evidence, yes, they do have physical dormancy. It applies for all the species, like soybean, all the peas.
Dry heat treatment in machine can reduce the dormancy period, question mark?
Can reduce the dormancy period. It can break dormancy. Yeah, it can break dormancy. If you dry heat them, that's kind of like an oven, right? You put the seeds into the oven, whatever that is, temperature. So if you have 70 degrees, you need a longer time for the seeds to break dormancy. But if you keep them for much longer, they would die.
Mm-hmm. From Gökhan Topaloglu. Hi, for wild eggplant rootstock seeds, they're seen dormancy. What can you say about light effects for breaking dormancy?
Oh, this is an interesting question, because I am now trying to understand if light has any influence in physical dormancy break. So what I have done some preliminary studies during my PhD and during my postdoc as well. So what I see is the day length, not necessarily the light, but the day length. Now I have done some other experiments. Summer temperature would break dormancy in Australia, in almost all the tropics. But you have to have the 12 hours of 60 degrees and 12 hours of that 30 degrees. That cycle has to be 12 hours and 12 hours. If you shorten the cycle to 1 hour 60 degrees and 1 hour 30 degrees, the seeds would not break dormancy even with that cycling. 60 cycles, 90 cycles, it didn't break dormancy for some reason. I I just couldn't understand why, and then I moved on. But that's a very good question. Actually, I'm going to spend some time about the day length time and the light as well. So for now, we don't know if light can influence physical dormancy break.
I got an awful lot of number of other questions here, but we'll ask one last one now. The others will go to you, Ganesh, and you can answer them later. What are the factors responsible for secondary dormancy? Interesting question, I think.
So for those who wanted to know more about my talk, or if you have any questions, you can send an email to me or to the organizers here. So to answer that question, this is something you can find more on this paper. Because of the only 30 minutes, I couldn't talk everything I want to talk about physical dormancy, but sorry, I'm going to quickly go there. It should be somewhere here. So it is for physiological dormancy. So what would happen is the seeds would have a dormancy at the time of dispersal, and then they enter into a state where they would lose dormancy, or they would become conditionally dormant, meaning they are dormant, but they can germinate. But if the right conditions didn't occur, they would kind of like re-enter the dormancy. So the conditions could be uncountable. It could be the day, the light, it could be the temperature, it could be the water availability, it could be the soil state, it could be how much sunlight, or it could be the burial depth. So all these factors would contribute to the development of secondary dormancy. But what I didn't know was if physical dormancy would go through the same process. And I believe even seeds with physical dormancy, meaning they are impermeable, they can cycle back to permeable state and then become impermeable. So this is again a controversial question, and some of the scientists don't believe that. But I demonstrated in this paper that that is possible, and there is a reason why that could be.
Thank you very much, Ganesh. That was a very nice presentation and very well received by the audience. In the top in the moment, we had 248 people who listened to your talk, which is a quite good number. And all the other questions which came in by now will be sent to you. And as you said, you can be contacted via your email. We are going to the next presenter now, and I would kindly ask you not to put during his talk questions to Ganesh, because it's hard to distinguish who the questions need to be answered by in a few cases. But I would like to introduce you to our next speaker. It's David Johnston. We just recently met at the ISTA annual meeting. David is from the US, and he has been involved in seed testing for over 35 years. While working in corporate, commercial, and governmental seed labs, he has experience testing a wide range of agricultural, turf, vegetable, flower, tree, and native species seeds. He's currently serving as the vice chair of the ISTA Germination Technical Committee. He's also a registered seed technologist accredited by the Society of Commercial Seed Technologists, and a certified seed analyst accredited by the Association of Official Seed Analysts as well. And of this organization, he is also co-chair of the Germination Committee, so a clear germination expert. And David, I would like to pass the floor to you to continue with your talk on dormancy breakage for germination testing.
Very good, thank you. Thank you, Ganesh, for that excellent presentation and all of the research you've done and your colleagues. That's amazing. And thank you to ISTA-ISSS for the opportunity to speak. And thank you for everybody that's tuning in and watching the presentation. So for sure, you could see with Ganesh's presentation that dormancy is a real challenge, is an understatement, especially for those of us involved in the world of germination seed testing. And some of us have even maybe considered a nightmare when it comes to trying to determine why the seed isn't growing. But if you've been in the seed germination testing business, you've experienced, you surely have experienced dormancy and the frustrations that it can cause. Let's see if I can get my slides to go. All right, as some professional information, it's already, as I was already in my intro, you can see that I just want to put those up there just so you can know a little bit about the person that's speaking, and just flip through these really quick. I've been in the seed testing business for a while and have experience testing a wide range of species, and not only have the perspective from the seed testing side as far as for seed companies, but also on the seed regulatory side, which is my current role, to determine seed quality and various things such as that. And just a disclaimer, I'm not a seed physiologist and I'm not a PhD, but I have friends that are. So in our world of seed testing, it pays to have a good network of seed physiologists and folks that are PhDs that are well versed in the deep meanings of seed dormancy, like Ganesh are and his colleagues. They can be an invaluable resource to you in your career when it comes to seed testing. So I thought I'd start out with a little poetry. That's the Little Plant poem by Katie Brown. It says, in the heart of a seed, buried deep, so deep, a tiny plant lay fast asleep. Wakes in the sunshine and creep to the light. Wakes in the voice of the raindrops bright. The little plant heard, and it rose to see what the wonderful outside world might be. But seeds are like some of us. We like to sleep late, and we're slow to wake. And so you get to this question of you wake up a seed when it is a late sleeper. So in my presentation, I'm going to give you a brief overview of what dormancy is and isn't, and the rudimentary requirements for germination, basic types of dormancy. Some of this is going to be an overlap, probably a bit of what Ganesh has spoken about so well. And so we'll move through those a little bit quickly. Just let you know some of the resources that are out there for all of us that are involved in seed testing. It's the handbook on seedling evaluations. We're going to hit that just a bit. And then the purpose of the dormancy breaking procedures that are in the ISTA rules, and kind of what their purpose is, and then what's the function of the dormancy breaking procedures, maybe a little bit of how they kind of what their purpose is, how they function, what do they do, and then how you have to report dormant seeds are in the ISTA rules. We call them fresh seeds. So if I use the term dormant seeds or fresh seeds, they're equivalent to each other. So when it comes to seed dormancy, it's basically it's the situation where a viable seed fails to germinate when it's provided with favorable conditions that should be initiating germination. This was in the, I believe it was in one of the ISTA book references, either in the rules or in the handbook. It stated that seeds must be imbibed and determined to be viable to be classified as dormant or fresh seed. Also, dormant is not the same as a hard seededness or state of quiescence. But these two conditions, hard seededness and quiescence, the seeds resting, they haven't imbibed any moisture yet. So in the information that was provided in the ISTA handbook, it mentioned that was one of the primary differences in those states of seed. Some of the basic conditions that typically required for seed germination are the appropriate moisture level, appropriate temperature, adequate air exchange, except for some species. When it comes to rice, it actually can germinate underwater, so the gas exchanges is a little bit different. There are probably other species, but my primary focus is going to be more on the cultivated type species. That's sort of an area that I have more experience in. And then exposure to light, whether it's natural or artificial, is required by some species, but not by others. So it can be a challenge to know what might be going on with the seed. Ganesh had mentioned this information that I had found was talking about the different types of dormancy. One was the primary type of dormancy, and this types they delay the germination of the seed. So you basically got a physical dormancy caused by the seed coat, or mechanical type of dormancy that may be restricting the seed from the embryo from cracking through and starting may imbibe water, but the seed did not allow the embryo to escape, you might say. And then there's chemical dormancies, might be found in the seed coat, morphological dormancy, physiological intermediate dormancy, or a deep dormancy, or a double dormancy, which we'll get into more on what these terms mean a little further on. But let's continue, because there's more basic types of dormancy. Could be that the seed coat prevents the seed from absorbing water. Seed coat mechanical dormancy, when the seed coat prevents the embryo from expanding and emerging from the seed coat. We experience this in some species that we test here, Paspalum notatum, or Bahia grass that we test here as a warm season grass that can have this big challenge. And then there's some chemical dormancies where there's certain chemicals in the seed coat or areas of the seed that prevent it from actually germinating. So there's different ways of mitigating those, and that's why I'm kind of giving you a rough idea of some of the types of dormancy, and then what I'm going to do is follow up on showing how the dormancy breaking procedures listed in the ISTA rules help to overcome, or try to overcome, some of these issues. So the morphological dormancy is when the seed embryo is immature when the seed separates from the mother plant. So it takes a bit of after ripening, or whatever term you'd want to apply, that the embryo has to continue to develop inside of the seed, even after the seed has dropped from the plant where it originated. I had experience with this. Practical experience was this when I had one lab I worked at. A person had some ginkgo tree seed, and we started trying to test the seed soon after harvest, and the seeds just would not germinate. And then we did some research and found out that it was one of the species that the embryo had to mature for a few months, I believe it was, after the seed had dispersed from the mother plant. And then we started testing germination, testing those seeds. I think it was maybe after like 4 months or so. Then the germinations were there because the embryos had developed, and we determined that a bit through some tetrazolium testing. And then the physiological intermediate dormancy occurs because the physiological conditions is not met. And then you can have a deep dormancy when seeds will remain dormant until favorable conditions are met. And then you can have double dormancy. So it's a combination of these other factors that are there. But you have to remember, dormancy oftentimes, it's viewed as a mechanism of survivability for the plant. If all the seeds don't germinate the first season, or 2 seasons, or 3 seasons later, that's their mode of trying to survive and continue to generate offspring into the future. But with the commercialization, or you might say domestication, of some of the species that are used in modern day agriculture, that dormancy has been kind of taken bred out of the genetics that the seeds are, maybe to help overcome some of that. Some of this, I think, was mentioned too, that Ganesh had mentioned about secondary dormancy. This can be caused by temperature extremes, whether your temperature the seed is exposed to is too hot or too cold. It can be caused by prolonged darkness or even prolonged light. That's the seed's way of knowing if it is in the right conditions to produce a viable plant. It's going to inhibit itself from germinating because it's sensing that the conditions aren't right. And then you can have, like, from water stress, it's too much or not enough, and then the lack of oxygen or gas exchange. Just a reminder that that these can occur because if you're in the business of seed testing in a seed testing laboratory where you're trying to determine the germination of a seed lot, your test conditions can actually induce a secondary dormancy because your testing temperature is too hot or cold. You're not providing the right amount of light or lack of light for the seed that you're testing, and the water stress can be too much or too little. The seed don't have enough water to imbibe and start the second phase of germination, and you're going to get seeds that will imbibe water and swell, but then they will fail to go into that second phase of germination by putting out a radical, putting out a root, or putting out a shoot. And then in a good example of this one here of lack of oxygen exchange, if you're familiar with the roll towel method in seed testing, where you place the seeds in moistened paper toweling in multiple layers, well, if you make your roll towels too tight, you're going to inhibit your oxygen exchange, and those seeds won't germinate due to lack of oxygen. So there's test conditions as a seed analyst you have to be aware of, that you don't see necessarily how many roll towels you can pack into a container because of efficiency. You have to be aware of these factors here, lack of oxygen. If you're a good recent example of this water stress, if you're one of the ISTA accredited labs or participant in the recent proficiency test using spinach, I'm also on the ISTA germination committee. We had a lot of questions about the seed germination on the spinach seed, and spinach tend to like a media that's a bit on the dry side. So if your towels were at that break point of being too wet, you might have seen some water stress and the seeds didn't germinate. So you might have been unfortunate. You usually get an A rating, and you might have wound up getting a C rating or the dreaded BMP rating, the below minimum performance rating, and had to go into some corrective actions. Just wanted to bring this to your attention. Hopefully most people on the call are aware of the ISTA handbook on seedling evaluations. There's a lot more information in there than just simply seedling evaluations. So section 2.9 specifically deals with physiological dormancy. It gives you not only this information, but it tells you how to carry out the germination test in the laboratory. And then it gives you some illustrated guides on evaluating seedlings for a large number of species. And it's an excellent supplement to the ISTA rules. So I would encourage you to go by the ISTA publication shop and make a purchase. It would be a worthy investment. It's a lot of information in there. A lot of work has gone into that document. And I would encourage you to go and make a purchase just for your own education and to see what's going on in that handbook. So when it comes to the germination test, of course, this is the objective, is to determine the germination potential of a seed lot, which can then in turn be used to compare the quality of different seed lots and also estimate the field planting value of that seed lot. So that everybody that's familiar with commercialized seed, and even for if it's just for research, the importance of seed germination and the rules that are set up to try to get these seeds to germinate. Germination of a seed in the ISTA test is the emergence and development of the seedling to the stage where its essential structures indicate whether or not it will develop into a satisfactory plant under favorable conditions in the field. So that's the ISTA definition and purpose of the germination test. It's not whether or not the seed will produce a radical or whether it'll produce a shoot or continue to grow, but it also must have essential structures that are considered favorable for the plant to be successful in the field. And so it's important to realize that for a germination percentage to be stated on a report of analysis or an ISTA orange or blue certificate, the germination is only the percentage of normal seedlings according to ISTA evaluation criteria that would go on to produce a favorable plant that would be productive. So that's kind of the frame of reference. And go on some more here. So in chapter 5, 563 says if you have issues where the seeds you've suspected some germination, then you can go into testing the seed using a combination or dormancy breaking procedures that are listed in the ISTA rules under these sections here. And for a retest, you can use any combination of these procedures, whether or not a specific dormancy breaking procedure is listed for that. Is listed for that species or not. It's just a matter of trying to get that seed to germinate and get the viability potential for that seed lot. So let's jump into some of the dormancy breaking procedures listed in 5631 in the ISTA rules. We have pre-chilling. Typically that temperature is going to be between 5 to 10 C for a typical pre-chill temperature. And then there's where what this would happen would be the seeds would be placed in the germination media that you're going to use for your germination test. So it'll be in a rolled towel or between blotter material or on top of blotter material or on organic growing material, whatever the approved media is for the test you're conducting. And then that seed will be put into a pre-chill chamber set at 5 to 10 C. So the seeds are going to be imbibing water while they're there. It's in the germination prep test, the seeds will be there imbibing water. It's not that you take your sample and then put it into a pre-chilling chamber at 5 to 10 C. It's critical that the seeds are placed on the moist media where they're going to be imbibing water at that cooler temperature into that chamber. So the seeds are there imbibing water. Now, when it comes to preheating, you're not going to have the seeds planted in your germination test media at this point. You're just going to take a submitted sample that you have, a certain portion that you're going to use for your germ test, and then you will place it into an oven to some place to dry the seed. Typically that's between 30 to 35 C, or for some species it's between 40 and 50 C. So the seeds are not moistened at this point. They're going in dry and they're going to be preheated. That's an important thing to know, distinction to know that. They listed in the section, it lists pre-storage, but really this is pre-storage is basically the seed that's in a suitable moisture level, at a suitable moisture level, internal moisture level, and it's placed into storage to allow the seed to rest. This state of quiescence, it's the seeds are not imbibed, just like preheating the seeds are not imbibed, whereas in pre-chilling, the seeds will imbibe. So your seed would be put into storage and allowed to rest. And so that's a common thing for a lot of species to have some level of quiescence innate into the seed that it will need to rest for a small period. Often in the agricultural crops, it tends to be just a few months, but in the seed world, sometimes we don't have a few months to wait. The company or the customer wants to know what the potential germ is strictly right after harvest, so we'll get into a little bit of that here in a minute. And then for some species, it needs to be exposed to light. That's a common thing for maybe the turf grasses, and also I'm familiar with lettuce. I worked in a vegetable seed company, and we always conducted tests using light for lettuce because it's some seed lots, some varieties, cultivars may require light, and some may not. So we would do a test, because the reason why we would conduct the test on the lettuce, we would conduct a test with the lettuce in the raw state, and then oftentimes lettuce is pelleted, so the seed is completely enclosed and it has no exposure to light. But in some seed lots and some cultivars, light was actually not required. So that's how we would screen which seed lots were suitable for pelleting, and then which ones may not be. They may have to have some other function done to them. But in the ISTA rules, it states a minimum of 8 hours of light per 24-hour period. And if you have a temperature an alternating temperature of 20 degrees for 16 hours and 30 degrees for 8 hours, you would be exposing the seed during your 8 hours of the high temp. So that's kind of some methodology there. One of the other procedures often for trifoliums would be to place your sample into a sealed polyethylene envelope, or basically a resealable plastic bag. The purpose of this is increasing the ethylene gas and the carbon dioxide for the seed to help promote germination. You can also, there's some species that in the rules for testing for breaking dormancy, say you need to use GA3 solution and then a potassium nitrate solution, and we'll get into a little more details on those. Potassium nitrate is a very common solution that's used as a dormancy breaking for brassicas and for a lot of the seeds that are in the Poaceae family to help stimulate germination. Another method to break dormancy is acid scarification. It's a means of trying to using sulfuric acid, but it's a means of trying to digest the seed coat a bit to make it more permeable to water. This can be kind of tricky because you're actually destroying the seed coat, and if you go too far, then it'll go through the seed coat into your seed and disrupt the embryo that's inside of there. So that's a tricky, that can be a very tricky procedure to use. And then there's mechanical scarification, where the seed would be chipped or nicked, or some way trying to penetrate just the seed coat enough that it will allow imbibation of the some of the dormancy breaking procedures that are not listed in 5631, but they are actually in Table 5A. If you're familiar with the ISTA rules, Table 5A parts 1, 2, 3 have the very specific germination procedures to use for different species. And you can see just in an excerpt from there, it gives you the substrate to use. All these terms here are defined in the ISTA rules in the germination section. And then it'll give you the approved temperatures to use. Anytime you see this symbol here of the two arrows pointing in opposite direction, that indicates a alternating temperature of 16 hours at the low side, 8 hours at the high side. And then you'll see some, I don't have a picture of it here, but it'll give you an example of some constant temperatures. It might be a constant 15 C or 20 C, just depends. And then over here, these are the days that you would count during your germination test. Gives you the duration of the test in the far right column. But this one here is where you would look if you're trying to see what are the recommended procedures for dormancy breaking. So anything that we're going over today, as far as approved dormancy breaking methods, would be found in column 6 for some of them. And then I think the trees and the shrubs have a different, maybe let me check my rules right quick, have a different column where you'll find dormancy breaking procedures for different ones. ISTA rules have species kind of broken up by it'd be still the same column 6 for trees and shrubs as well. They haven't broken up by different types of seed or groupings of seed. Some of the other dormancy breaking procedures that are not found in table, but they are in Table 5, would be pre-washing the seed, pre-soaking the seed, and these are just a few that are ones for pre-soaking. These are to help essentially try to remove any germination inhibiting chemicals that might be in the seed coat of the seed itself. Try to leach those out so the seeds can germinate, similar to what they would do in nature, where they would be planted and then the rain would leach out these germination inhibiting chemicals. One of them might be removing the enclosing structures that is in the seed. And a good example of one where if you had a sample of peanut seeds or ground nuts, depending on what your common name is for them, you would take those seeds out of the pod instead of planting them. Doing your germination test with the seed still in the pod, you would actually take the seeds out of the pod. The enclosing structure is considered. And for some of the tree seeds, the recommendation is to incubate the seeds in a moist substrate, and that's all defined in column 6 as far as which one you would use. Actually, alternating temperature during the germ test is considered another dormancy breaking procedure that causes the seed to help. There is a mention of ethylene gas, but there's really no procedure provided. But this was just mentioned in the handbook dealing with helianthus and rice, sunflowers and rice, or rhiza. But there's really no procedure given, just sort of a heads up on that in the ISTA germination committee, we're trying to schedule a use of ethylene for sunflower to break the dormancy. Freshly harvested sunflowers, the helianthus, can be a real challenge to germinate. Some of the labs that are familiar with them believe that seed has a double dormancy, so they're trying to make that figure out how to break that dormancy for freshly harvested seeds. So when it comes to dormancy breaking, some treatments are before the germ test and some would be after the germ test. So like the pre-storage or pre-chilling, you're going to incubate that seed in a cool, cold, moist substrate. So that would be done before the germination test. If you preheat the seed, you're going to be putting it under those extreme heat conditions before the germ test. And then if you're going to pre-wash the seed or pre-soak it, of course, that would be done prior to the germination test. And then removal of any of the enclosing structures would be done before the test. And then if you're going to do any type of a scarification procedure, that would be done before the test. So these are like prepping the seed prior to your germination test. It's kind of like I mentioned earlier, you need to be cautious if you're going to be using the mechanical scarification and the acid scarification, that you're not damaging the seed beyond the level of the seed coat. So be careful which end of, if you're doing a chipping or fracturing of the seed coat, make sure of your seed morphology that you're not actually going to mechanically scarify the area where the little radical is at, or where you're going to be actually damaging the seed prior to your germ test. It's going to skew your germination results. So just a word of caution on these two procedures. They can be a bit invasive. So the treatments during your germination test, these are maybe kind of obvious for us all, but of course, if there's light, you would be exposing that seed to light during the test. Your alternating temperatures, that would be your test itself. If you're putting the seed into these envelopes or Ziploc bags, of course, that's when you would plant your seed on your media, and you just slide it into the Ziploc bag and close it. You wouldn't want to collapse the bag. You might allow it to pillow a bit just for that gas exchange. So you don't want it to be completely collapsed and suffocate the seed inside. You do want to give it a little bit of airspace in there to allow the seeds to have air and get the gas exchange. And then, of course, with the gibberellic acid, this is what I probably should have added another comment. I believe that some seeds you add this to the germination substrate, and then I believe there's a reference to maybe a species or two where you would actually soak the seeds in GA3 prior to your planting the seed on your media. And then on the potassium nitrate, that one usually is the same as you're moistening your test substrate with potassium nitrate to help overcome break any dormancy. Just a kind of an observation that we've noticed on potassium nitrate is you want to be cautious when you're using that on your media, that it at times it can cause some of the root tips to get brown, and it can cause some. It seems that at times also, if your seed has a lot of pathogens on its seed coat, this can sometimes kind of enhance that a bit. So just a word of caution on some of the potential, I guess, side effects of potassium nitrate, that just give you a heads up and be aware of it. So like I mentioned earlier, here's an excerpt of Table 5A, Parts 1, Part 2, Part 3, and it specifically will tell you which recommended dormancy breaking procedures are allowed. KNO3 and pre-chill you'll see is a very prominent dormancy breaking procedures for a lot of species. And then you can see here with the trees and shrubs, you might have a soak in the water. And here's another one where it talks about cutting off a scar into the seed, or where you're telling you to do certain things, you know, specifically to that species that you would find over in this column. And then the one for herbs, spice, flowers, herbs, and spices. It's pretty common for a lot of flowers, I think, that need to be exposed to light. So sometimes has to do with the seed size in general. I mean, this is not a fast rule, but in general, your smaller seeded species, whether it's in the poaceae, the grasses, and maybe some of the flowers, it's pretty common for them the small seeded species to need to have light exposure. So that's something that just a general rule of thumb, but that's where you find that information in the ISTA rules. And just in general with the ISTA rules, all of these methodologies for different species are based upon some validation study or such things, and these are constantly under review and can be changed. There is a process. It's the rule proposal process where you conduct a validation study through the germination technical committee to help that, and the statistical committee to help you set up a validation study to change or add a species. All those things can happen. These aren't hard, fast rules. They are what we use because they are approved, but these methods can be changed. So let's jump into what's the function of the dormancy breaking procedures. So part of their purpose is to simulate the cooler or cold winter conditions that often break dormancy in nature. So you can see, you know, some of these, that's what their purpose is to help kind of recreate what happens in nature, where the seed may be dropped to the ground, and then over the winter conditions, the dormancy is broken, and by springtime, the dormancy has all been mitigated through the natural climate and conditions, and it's ready to germinate whenever the conditions are right in the springtime. Then you can artificially, it decreases inhibitors and increases promoters inside the seed, is some of the dormancy breaking procedures. That would be one like where you're doing the pre-wash soaking, and then you're increasing the inhibitors, whereas this one would be an example of the seeds that are placed into the sealed polyethylene envelopes, where you're increasing the promoters inside of the seed, or you're adding GA3 or Cano3. This one here talks about you're disrupting the seed coat to allow for imbibation and gaseous exchange. So that example of that again would be the scarifications, where you're trying to do that. And of course, you're stimulating germination with exposure to light. So these are what kind of the purpose, the function of these dormancy breaking procedures. With the alternating temperatures, you're trying to simulate a day-night cycle, and that can stimulate seeds to germinate. Stimulating internal activities, maybe by adding some of the germination promoting chemicals to encourage germination. And this is the one for more the sealed envelope situation here, where you're increasing the ethylene, and that naturally the seed will give off in the carbon dioxide to try and get the seed to germinate. Removing other structures is another way of doing that. So there's, like I mentioned, there's more details in the handbook on sealing evaluation. It's a viable resource. Gives you much more detail than the ISTA rules. That's not really the purpose of the ISTA rules in a sense. It's more of this handbook is more where you're going to find a lot more detailed information. Just a reminder again that if you're in the lab and you're testing your seed, your test conditions can cause dormancy, secondary dormancy. So always be aware of that. Something to always be cautious whenever you're doing a germination test. Always be aware of your test conditions at all times, and try to mitigate things that are there, and never be hesitant if you believe your test conditions have caused a problem. Always retest it when you're in doubt. If you don't, you can report inaccurate results. It could be for some of the reps, or it could be maybe one or two of your replicates had a problem. And the ISTA rules has a tolerance table to compare replicates to make sure they're all within tolerance of each other, statistically based. So that will tell you, that'll give you the level of confidence if you see a wide variation between your replicates. Use the ISTA rules tolerance tables for comparing replicates to help make that decision. There's no need to retest if statistically your replicates are within statistical tolerance. Let's see. This induced dormancy can also be greatly inconsistent. It may vary by species, by seed lot, age of the seed lot, test method, test replicates, etc. So always be aware of those things. Always remember, just because a seed does not germinate during your germination test, don't assume it's automatically dead. It's dead. And so that kind of will lead us into, let me get my notes, into the next idea of, okay, so you're telling me that if the seed doesn't germinate, then it doesn't automatically mean that it's dead. So Mr. Presenter, how am I going to find out if that seed is dead or not dead? Well, there's a chapter for that. It's called the biochemical test for viability of the topographical tetrazolium test. Hopefully many of you on the call are familiar with this test, but I'm going to give you a quick overview, because you're now going to have a tool to help determine if a seed that did not germinate but imbibed water is dead or alive. So the objective of the tetrazolium test is to make a quick estimate of the viability of seed samples in general, and showing if they're dormant or if they didn't germinate, because regardless, if the seed did not germinate during your germination test, then it's possibly dormant or dead. And if it's dormant, it's still viable, which means the seed is still respiring. The tissue is still respiring. That under certain conditions, that seed would probably go ahead and produce a seedling, which is a good thing, because now we can tell if the seed is not viable, then the tissue will not. We can determine if that's the case too, and I'll show you here in a second. So this tetrazolium test is a tool to use after the germination test, which we would refer to as a post-germination TZ test, or it can be used as a standalone test, where you do it with you would do it in lieu of or in addition to a standard germination test. The seed would be allowed to be imbibed with water. So during your germination test, the seed are already imbibed with water. But in a standalone TZ test, you would have to actually imbibe your seed with water and then go through this procedure to see if the seeds are viable. It's a quick test, usually takes maybe 2 to 3 days, maybe longer for some recalcitrant type species are hard to really dormant species. But the thing you need to know, if you're using the TZ test as a standalone test, it will not tell you the level of dormancy. So that's something you need to be aware of. It'll tell you the potential viability, but it will not tell you the potential dormancy in the seed you're testing. The germination test will give you that information about the potential for the seed to germinate. So you'll produce seedlings, but then the ungerminated seeds, that is where you will determine the percentage of viability. So the basic process for the TZ test is going to help you determine the viability of these ungerminated seeds. So the seeds are already been imbibed on your germination test, and you're going to carefully cut the seeds, allowing the embryo portion of that seed to be exposed to a tetrazolium solution, and then any of the respiring living tissue will stain red in color. So that's the basics. So after the appropriate amount of time, and this can vary, you'd have to look at the ISTA rules in chapter 6 to see the tables there that talk about possible or approximate staining times. You'll have to then interpret the staining patterns of the embryo and the tissue that's there. Then there's guidelines for that. And after you've evaluated each seed, you're going to determine if the seed would be considered viable or non-viable. And sometimes you might see some that are questionable, and sometimes that means you need to stain a little longer, or you can't determine if they're viable or not. But the ones that would be considered viable are then reported on your ISTA certificate as in the fresh seeds category, and the non-viable seeds then would be reported as dead seeds. So I just gave you this example. Picture's worth 1,000 words, so here's your 1,000 words right here to hear me talk. So what you're going to do, this is an example for cereals. So this is the embryo section, and this is endosperm. Same here, embryo and endosperm. Is on this one, you're going to do a longitudinal cut right through the embryo, right this way. On this method, you're actually just going to cut it off here, and then your TZ solution will go through this cut you've created here, or it'll go through this cut that you've created here. So you would discard this part of the seed, but you would keep this part and put it in your TZ solution. In this method, you would slice it through here, and you would leave the seed connected and intact in both halves, and then you would evaluate these halves here on both sides of the embryo. This one you're going to have to maybe gently tease the embryo out to evaluate if it's staying properly.
David, just to let you know, we exceeded the time nearly. If you want to have one or two questions, then that would be fine to finish up.
All right. Do we just to finish up quickly?
Not to hurry, but to do it a little bit more quickly.
Sure, I'll do it. Just about finished. There's only a couple slides left. So this is where you would see if you did this, cut the seed longitudinally, this is the embryo that you would see. And then you can see these would all be considered normal seeds, even though there's not 100% staining. And then this would be one that's cut transversely. So you would see here how you evaluate it, but these would all be considered viable. And here's an example of some non-viables where it didn't stain the shoot, didn't completely stain the root. This one would be completely dead because it didn't have any staining at all. So the ISTA rules would show you more detail on how these are done. And then I'll just quickly run through this. This is basically about reporting your test information on the ISTA Orange certificate. You have to state any special methods that you used and any under the other determinations section of your ISTA certificate. There is a requirement about if you suspect 5% or more fresh seeds, then you have to do a viability check. If it's less than that, you don't, but it's an option. Another good option is to do a parallel test where you're using you'll conduct two tests at the same time, one with germination or promoting procedures and one without, and then you report the best. So it's important that you make it known. That's the challenge, is making it known which if you use the dormancy breaking, you're actually could be masking the dormant seed percentage. But with a paired test, you're going to be finding the level of dormancy. So this is the other seeds area where you put your other determination area, where you put your what dormancy breaking methods you used, and then this area is where you'd report your fresh seeds that would be considered dormant seeds. And this is a reference of the material that is used. Don't forget, 100-year anniversary for ISTA coming up in 2025. If you'd like to contact the Germ Committee, you can punch that little QR code and take you right there. All right. Thank you for your attention. Appreciate it. And there's the contact information. You're welcome to shoot an email to me or to the Germ Committee. Jillian is the Germination chair. I'm the vice chair, so we're happy to entertain any questions you'd have.
Thank you very much, David. Maybe time is over, but we have I have a few questions for you. We cannot answer all of them. But,
yeah, please forward them to us. If you have them, just shoot them by email. We'll be happy to.
Yes. One interesting one which I found here from Natalie was, do you know if dormancy can come back after a while?
Well, I know personally have experienced that with quo a pretense with the Kentucky bluegrass, that research within AOSA and SCST have shown that, just like I think Ganesh even mentioned too, is that this dormancy can cycle in and out. And so, yeah, I would say that it can come back into the seed and cause you some real problems. So you always have to be aware of that. So I think it can be cyclic.
Okay. Next question, please elaborate table 5A. Potassium nitrate and pre-chill together or either of them?
Oh, that's a good question. Typically, I've always done them together. I've used both. Either one's allowed, but I think you can use them both together. Yeah, I don't think there's a restriction on one or the other. I mean, it's kind of like I'd referenced earlier. It did talk about combinations of pre-chill dormancy breaking. Excuse me, using combinations of approved dormancy breaking procedures. So yeah, you can use them both together.
Okay, and why alternating temperature is more effective in analysis of seed germination?
Why is it more effective? Is that the question?
Yeah, that's why. Yeah.
All I can say is that the methods have been validated through validation studies, and so that was proven that the alternating temperature produced a more accurate germination potential result than a constant temperature. But I don't have this. You know, I'm not the one that developed the methodology on those. That's what I would say it's been determined via validation studies.
Okay,
Physiologically,
and then
what's happening? Maybe you know, Ganesh can answer that part about that breaking of that. But yes,
I have a last question for you as well. Do ISTA rules specify the light quality and quantity for the dormancy breaking? I think that is very interesting.
I don't have the reference right in front of me, but yeah, it does mention the. We used to use foot candles, but now I think it's changed. But there is a recommendation, I believe, is the wording that we use, not a requirement per se, but a recommendation. In the ISTA Germination Committee, we have numerous discussions on that topic about light quality. How do you measure it? Where do you measure it? I mean, when you're thinking in a practical sense, if I'm putting seeds within a media, rolled towel, for instance, and there's a light requirement, or if I'm putting a germinator and filling it full of species that require light, how am I going to make that light equivalent throughout that whole chamber? And it's just not practical. So it does specify more details in the ISTA rules, and I think it's in the ISTA rules, or at least in the handbook, about the light quality. And so I would have to refer to that for you. If you want more detail, you can shoot me an email so I can answer you more directly. But it does specify it in there.
Yeah.
But it's a recommendation, I think. Yeah.
Thank you very much. All the questions which were not answered, we will send also to the presenters. So what you find then is that they will you can get the answer directly. And if you have more questions to them, David has got his email address here, but we also have the email address of Ganesh. So what I did in the meantime, I put in the chat for you the link to the ISTA YouTube channel. You can use that link. Ganesh, would you open your camera as well, just for the final
final farewell?
Final farewell, yes.
I'm trying, but I should know how to do that actually. There is a webcam. Oh, I see. Okay, I think, yeah.
You did it. Good experimental scientist. Thank you very much for all of you. That was this ISTA webinar, ISTA-ISSS webinar. If you need more information about the organizations, you can visit their websites. And as I mentioned, it's all on the ISTA YouTube channel. During the course of the week, it will be there. Thank you, Ganesh. Thank you, David, for your presentation. We had very positive feedbacks in the background. And I close this session for today. And the next will be in about half a year. The next ISTA-ISSS webinar, and we see you again next time. Thank you very much, and have a good day or night or wherever you are in the world. Thank you very much.
Thank you so much.
Bye.
Thank you, Andreas. Thanks. Good job, Ganesh. Thank you.
Likewise, David. Very nice.